Hi everyone, my name is Julia Casorso, and here I will be presenting my research on the evolution of alcohol metabolism in the human lineage. Sometimes simply referred to as alcohol, ethanol is the active ingredient in alcoholic beverages that causes drunkenness. The ethanol in your drink comes from a process called ethanol fermentation. This is where yeasts convert sugars like glucose into ethanol. Humans began using this process to intentionally ferment alcohol during the Neolithic Revolution 12,000 years ago. But ethanol fermentation also occurs naturally in fruits and nectars. The ability to digest ethanol varies widely among the animal kingdom. What's interesting is that humans and African great apes, so gorillas, chimpanzees, and bonobos, can digest ethanol 40 times more efficiently than most other mammals. This ability comes from a shared mutation in our DNA that causes enhanced activity in an ethanol metabolizing enzyme. Researchers have hypothesized that our most recent ancestor we share with African great apes got this mutation and passed it on to its descendants. What I want to know is, why was this mutation passed on? Was it random chance? Or did it provide our ancestor with some sort of survival advantage that allowed it to pass the mutation on? The dietary exposure hypothesis suggests that increased exposure to ethanol in ripe fruits and nectars was the driving force that favored this dietary adaptation in our last common ancestor. This hypothesis has two main predictions. First, it predicts that in Africa, where our last common ancestor may have lived, fruits have higher ethanol content compared to fruits in other primate habitats around the world. Second, it predicts that ripe fruits that fall off trees are likely to be more fermented and contain more ethanol. Our ancestor may have been transitioning from a life in the trees to a life on the ground and would therefore come into contact with these fallen fruits more frequently. The focus of my research is to test this hypothesis by gaining an understanding of ethanol concentrations and sources of variation in ethanol in wild fruits eaten by primates around the world, something we currently have a poor understanding of. To measure ethanol in fruits, we have been using a newly developed ethanol analysis system. The first step is picking fruit. So far, I have collected commercial fruit from the local grocery store, and my colleagues have been collecting wild fruit at a site in Costa Rica where we study wild capuchin monkeys. Next, we incubate the fruit inside of a plastic bag. We then test the air within that bag with a breathalyzer, exactly what a police officer would use to test someone's breath if they were pulled over, which we have modified for testing fruit. So far, we have found that 57% of the 14 commercial species tested and 65% of the 65 wild species tested produce ethanol, mostly when ripe and overripe. In the bottom graph, you can see that average ethanol levels are relatively low in both commercial and wild fruits. However, ethanol production ranges much higher in commercial fruits, and we found that commercial fruits had higher ethanol levels at all ripeness stages. This may indicate that ethanol production has been influenced by fruit domestication or by the crop harvesting and storage process. But the most interesting thing we've found is shown in these graphs where we have the ethanol production of individual peaches and guavas as they transitioned from unripe to overripe, with average ethanol production indicated by the solid line. You can see that ethanol production shows a clear trend over time, where it begins at 0%, peaks once or twice as the fruit ripens and yeasts convert sugar to ethanol, and then trails back down to zero as yeasts begin to consume ethanol once sugar levels get very low. Now, primates are very sensitive to ethanol odors and are often observed sniffing fruits before eating them. So, primates might use this change in ethanol odor to identify the ripeness stage and sugar content of fruits to optimize their foraging decisions. The take home message is that we are finding ethanol present in the majority of ripe fruits, and ripe fruit is a really important food source for primates. This means that our findings so far are in support of the dietary exposure hypothesis, or the idea that our efficient ethanol metabolism originally evolved as an adaptation for exploiting ethanol producing fruits. Overall, this research marks the start of a growing global dataset on fruit ethanol and primate foraging behavior, and it will also be important for comparative studies with other fruit eating mammals to help us understand dietary adaptations more broadly.